Proteins that are packaged into transport vesicles that will bud off of the surface of the rough endoplasmic reticulum will then travel to different destinations in the cell. But many of those transport vesicles will carry the proteins to an organelle called the Golgi apparatus. The Golgi apparatus, like the ER, is an extensive network of membrane. And that membrane is also organized into flattened sacs. But these sacs have a very different appearance to them, as you'll see in just a minute. The Golgi apparatus is typically located near to the rough endoplasmic reticulum because, again, many of the proteins leaving the RER are going to come into the Golgi, and they enter the Golgi for further modifications. The Golgi apparatus actually has directionality. In other words, one side of the Golgi is different from the other side of the Golgi. One side is going to be facing the ER. That is referred to as the cis face of the Golgi. The other side is going to be facing away from the ER towards the plasma membrane of the cell, and that's referred to as the trans face of the Golgi. We talk about these two sides of the Golgi, the cis and the trans face, because those sides are involved in the direction of transport through this organelle. Proteins are going to enter through the cis face and then will travel through the Golgi apparatus and exit through the trans face. Now, just like we see in the ER, proteins that are ready to leave the Golgi apparatus will be packaged into transport vesicles and will bud off of the surface of the Golgi apparatus. Compounds that leave the Golgi apparatus in these membrane-bound transport vesicles can travel to different locations in the cell. They essentially deliver this protein to its final destination in the cell. Some of the transport vesicles carry the protein to the plasma membrane, and from there it will either be secreted from the cell or used in that location in the cell. So what exactly is going on in the Golgi apparatus? Just like we saw in the ER, the Golgi apparatus is an organelle that is involved in processing protein. The Golgi specifically performs the work of modifying proteins with carbohydrate. Small chains of carbohydrate will be added on to protein in the Golgi apparatus, creating what are referred to as glycoproteins. Interestingly enough, complex carbohydrates are also synthesized inside the Golgi apparatus. On this slide, we can see a drawing of the Golgi apparatus. You can't see it in relation to other structures in the cell, but the artist has labeled this as the cis face and this as the trans face. So you have to imagine this organelle in 3 dimensions. We have cut through that organelle to produce this particular view of it. You can see that it is a network of membrane that is organized into individual compartments that are flattened like sacs. You can also see that all around this interesting looking organelle are these small membrane-bound circles or spheres that we refer to as transport vesicles. There are vesicles coming into the cis face and there are vesicles leaving the transface. These vesicles, which are hollow, contain proteins that are coming out of the rough ER. Remember, the nucleus, the rough ER, and the smooth ER are all continuous with each other. Their membrane is connected, but the Golgi is not connected. So in order to deliver things from the rough ER to the Golgi, we need these little transport vesicles to do the work. These vesicles are going to come up to the membrane on the cis face, and they're going to merge with it, allowing the proteins to enter into the cavities that are within the network of the Golgi apparatus. The proteins will then travel through the compartments, get their modifications made, and then they will be packaged into other vesicles that will bud off of the surface of the transface and go to whatever final destination they're going to. As I said before, the Golgi apparatus has a very unique look to it inside actual cells. Now, if you take a look at this EM image, this is a transmission electron microscope image, you can see in the center the Golgi apparatus. Now, it's not very easy to tell where the ER is in this image, so we will just focus on what the operator has labeled as the cis face and the trans face. So we know the rough ER is up here somewhere because the cis face is going to be oriented toward the rough ER. And we know that the plasma membrane is down here somewhere out of our view because the trans face is going to be oriented towards the plasma membrane. These are the flattened sacs of membrane right here. And as you saw in the picture a minute ago, these sacs tend to start small, then get larger and larger and larger, and then get smaller and smaller and smaller once again. The Golgi apparatus has that very unique structure to it. The sacs start off small, then they get longer and longer and longer, and then smaller and smaller and smaller. And they're lying right on top of each other like flattened pancakes. But I think what makes the Golgi really unique visually are these. These are all of those transport vesicles. The white that you see inside of these circular structures, that's the space inside of the vesicle. That would be where the protein is located. The vesicles are coming down and entering the cis face by merging with the membrane of the Golgi apparatus, and proteins are being packaged into vesicles that are then budding off of the transface and traveling to other destinations in the cell. There are two other organelles that look an awful lot like transport vesicles. They are small, they have a single membrane around them, and they're spherical. One is called the lysosome, and one is called the peroxisome. So we often describe lysosomes and peroxisomes as being vesicular organelles. They look like vesicles. They look like spherical compartments that are within the cell itself. They're small compared to the other organelles, and they all look the same. So you would be hard-pressed to look under a microscope and tell the difference between a transport vesicle, a lysosome, and a peroxisome. They're all going to look very similar. They look like small, spherical compartments. Their function, however, is very different. We already said that transport vesicles have the job of carrying things from location to location, primarily proteins. Lysosomes have a very different function. Inside of lysosomes, there are a type of enzyme referred to as acid hydrolases. Now, this is a big family of enzymes, and like the name suggests, when the pH is acidic inside of the lysosome, these are enzymes that can hydrolyze or break apart biological molecules. It's the job of the lysosome to break down almost any biological molecule that comes into it. And the purpose of breaking it apart is to recycle the components of that biological molecule. So you can think about lysosomes maybe as being the kind of digester organelle that the cell has. Remember, all of the molecules that are inside of a living cell have a limited lifespan. So there will come a time, for example, when certain proteins are finished. They're done doing their job. And rather than just degrade them and let them go to waste, the cell sends them to the lysosome so that they can be digested. They can be broken apart into their amino acid subunits and reused, recycled in the cell. There are other things that get sent to lysosomes as well, particularly infectious things like bacteria. There are certain types of bacteria that can get into cells. There are also viral particles that get into certain types of cells. And one of the ways that the cell degrades those invading infectious pathogens is by sending them to the lysosome. Now, acid hydrolases are dangerous things, and you wouldn't want them just floating out in the cytoplasm of the cell because, as we said, they can break down almost any biological molecule. So by housing them in the lysosome, the cell is keeping them safe. The cell is preventing self-digestion, if you will. If you examine the inner surface of a lysosome, it's actually coated with a special kind of carbohydrate. And that's important because that prevents the acid hydrolases from digesting through the wall of the lysosome. If you take a look at this EM image down at the bottom of the slide, you can see some lysosomes here, 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 and here. You can see that they are different sizes. That's quite common. And you can see that there's different shades of gray inside the lysosome. The color that you're going to see inside of a lysosome is going to vary. And that's because each of these lysosomes is at a different stage of digestion of whatever is within it. Peroxisomes have a similar job to lysosomes in that they are in the business of degrading biological molecules. But peroxisomes are different in that they're going to break apart different materials than what the lysosomes are going to break apart. Peroxisomes are vesicular, as we said, spherical organelles that are surrounded by a single layer of membrane. Peroxisomes got their name because they are capable of degrading a molecule called hydrogen peroxide. Peroxisomes can actually create or generate hydrogen peroxide as well. Hydrogen peroxide is toxic to living things. Hydrogen peroxide can be converted to water and oxygen with a special enzyme called catalase. So, anytime a cell uses oxygen in its metabolic processes, there's a risk of that oxygen becoming hydrogen peroxide. Hydrogen peroxide is toxic to all cells, both eukaryotic and prokaryotic. So a cell has to have a way to deal with any hydrogen peroxide that is generated. And that's the job, again, of this catalase enzyme, to break it apart and detoxify it. Now, a lot of research goes into the function of peroxisomes even today, because there are some scientists who believe that peroxisomes help regulate all forms of toxic oxygen molecules in a cell, including oxygen radicals, which are believed to play a role in aging. So scientists that are involved in anti-aging research are particularly interested in studying the function of peroxisomes. Now, peroxisomes don't only break down hydrogen peroxide. There are other molecules that they're capable of degrading as well. Those include things like methanol, ethanol, formaldehyde, and other similar chemicals. Peroxisomes can also degrade unusual forms of amino acids, unusual isomers of amino acids that the cell doesn't have a use for. And just as we said with mitochondria, the number of peroxisomes that you will find in a cell is going to vary depending on the type of cell. So cells that encounter a wide variety of potentially toxic molecules that need to be broken down are going to have an abundance of peroxisomes. So a great example of that would be liver cells or hepatocytes and kidney cells. In the image over on the right-hand side of the slide, this large circular organelle that you can see right here, this is a peroxisome. Again, peroxisomes are going to be very difficult to distinguish from lysosomes and from other transport vesicles. The coloration that you'll find inside the peroxisome is going to vary depending on what chemicals are located in the peroxisome at that moment and what stage of digestion is the peroxisome is at. Now, I want you to notice that in this particular EM image, you can also see another organelle that we talked about right here and right here. This happens to be Golgi Apparatus. You can tell that it's not ER because if it was rough ER, there would be ribosomes on the surface of that organelle, and those are not visible. You can tell that it's not smooth ER because it looks too much like flattened sacks of material and not like that tubular network that we looked at.